Hello and welcome to this course. I'm Dr. Rana Wafai. I have a PhD in biochemistry, and in this brief video I will introduce you to this course about amino acids and proteins. Now this course is intended for any student with a bachelor degree or a master's degree or a basic knowledge in biochemistry who seeks to improve his knowledge about amino acids and proteins. It is also good for those who are already familiar with amino acids, with proteins, but who are interested in learning more about the protein structure and function. Now, in this course, we are going to cover several range of subjects, including structure, properties, and classification of amino acids, how amino acids can act as acids and bases, and therefore they can even act as buffers, the levels of protein structure ranging from primary, secondary, tertiary, and quaternary structures, the peptide and the protein conformation, the peptide bond conformation, as well as the different conformations of proteins. How do proteins fold? We are going to briefly talk about protein functions and the concepts in protein function, including reversible binding, the effect of ligand binding, the effect of allosteric modulators. An example of the protein that we are going to take to describe the reversible binding of a ligand to a protein are hemoglobin and myoglobin. So first we're going to describe the binding of oxygen to the heme molecule, which is present in both of these proteins, myoglobin and hemoglobin. We are going to describe the kinetics of ligand binding to a protein, taking first myoglobin as an example, where we're going to explain fully the kinetics of oxygen binding to myoglobin. Then we're going to talk about the cooperative binding of oxygen to hemoglobin molecule and how this differs from myoglobin. So we're going to differentiate between the two proteins, myoglobin, which is a protein formed of only one polypeptide, and hemoglobin, which has a quaternary structure. It is formed of 4 subunits and which binds oxygen with cooperativity and which is considered to be an allosteric protein. And finally, we are going to discuss some of the allosteric effectors that affect the binding of oxygen to hemoglobin molecule to modulate its affinity. Now, after taking this course, you will be able to identify the different types of amino acids, their properties, and their classification. You're going to be able to describe the role of these amino acids as acids and bases, identify and describe the different levels of protein structure. In addition, you're going to understand the peptide bond structure, how proteins are going to undergo folding into their native conformations. You are going to be able also to list and understand the different concepts related to protein function. Two proteins that are given here as examples are the myoglobin and the hemoglobin. We are going to study, you will be able to determine the kinetics of oxygen binding to heme, and to differentiate how this binding occurs in myoglobin as compared to hemoglobin. And finally, you are also going to be able to explain the effect of allosteric modulators on allosteric protein, given of course hemoglobin as an example, where we're going to discuss the allosteric effectors that affect the oxygen affinity and the binding to hemoglobin. I hope you will enjoy this course and you register for this course, and in case you need any help regarding any topic in biochemistry, we are here to help. Thank you.